Hello and welcome to Cornell Keynotes. I'm very excited to be hosting the Good Dogs, Great Data keynote of 2026. We'll be discussing dog genetics and genomics. Today we're going to be talking with Cornell faculty about an exciting summer course that they've put together called Genomic Data Science in Dogs. My name is Anthony Hay. I'm an associate professor and director of undergraduate studies in the Department of Microbiology at Cornell in the College of Agriculture and Life Sciences. Joining me in the studio today are the Genomics Data Science course developers, Dr. Adam Boyko, associate professor, Dr. Jacquelyn Evans, assistant professor, and Dr. Jess Hayward, senior research associate at Cornell University's College of Veterinary Medicine in the Department of Biomedical and Translational Sciences. Thanks so much for joining me. I'm sure everyone would like to hear a little bit about you and your background, so why don't you take turns and introduce yourself?
Sure. I'm Adam Boyko. I'm an associate professor in biomedical and translational sciences. My lab's research focuses on canine genetics, which is everything from the domestication of dogs and the population history of dogs to the genetics that underlie all of the behaviors and traits and morphology, as well as the diseases that we see in dogs and understanding the genetic and environmental basis of that. And then building genomic tools that we can use to investigate dogs further, including genetic testing tools that owners can use and veterinarians can use and also breeders can use, and whether we can improve the way we breed dogs with genetics.
Awesome.
Hi, Anthony. I'm Jacquelyn Evans, and my background is in canine genetics. My research is focused on understanding what are the genetic variants that drive diseases in dogs, and I'm particularly interested in complex diseases like cancer and autoimmune diseases. These are later onset, and they usually involve multiple genes as well as environmental risk factors.
That really resonates with me. My mom's adopted, so I was really interested in doing 23andMe many years ago for the same reason, trying to figure out what my disease risk was. So. All right, Jess.
Hi, yeah, thanks. So my name's Jessica Hayward. I did my PhD at the University of Auckland in New Zealand. And I was studying the DNA sequences of viruses, so specifically cat and dog viruses. And then about 15 years ago or so, I switched to canine genomics. And that's what my main research focus is. More recently, it's also been, I've branched out a little bit into both the feline and the equine model. And so I'm particularly interested in complex diseases. I'm also interested in looking at using cell-free DNA as a biomarker, especially for cancer.
Oh, cool.
Yeah, and also in the studio with us today we have Blueberry. This is my dog, and he's, as you can see, he's a 3-year-old golden retriever, and he loves green beans. So he is a dog. He's a golden retriever, so he is food motivated, and he loves people. He has to meet all the people, and he enjoys swimming.
Great. Oh, great, thanks. Before we dive into more details about the course itself, let's talk about canine genomics and what it actually is and why we'd be interested in it.
Yeah, so canine genomics is a study of dog DNA. And so dogs were the first domesticated species. So 20,000 years ago or so, the first wolves became the first dogs. And that was a genetic process. It changed the DNA sequence. That's what the difference is between dogs and wolves. And then the dogs hanging around the people, we started to utilize them for different tasks and jobs. And so we started to diversify and breed dogs. And now they're the most diverse land mammal in terms of phenotypes. So the difference in size between a Chihuahua and a Great Dane is about 50-fold. So more than any other mammal species that I can think of. And it's not just body size, it's body shape, its coat, and its behavior, and its temperament, and all of these things that we've bred for, and makes dogs, and that's all in their DNA. And so it makes them super interesting to try to study what are the genes that affect these sorts of complex behaviors.
I'm guessing we never bred them for disease resistance, but I'm assuming that there's differences in the different breeds.
You can see different disease risk profiles in different breeds. We can even see that just as some human populations have evolved malarial resistance, we see some dog populations that have have different resistance to different pathogens in their environment. So evolution clearly has been taking place, not just us artificially selecting specific breeds, but also natural selection in the environments that they settled in.
So that sounds like part of what makes them a powerful model. Are there other aspects that make them a powerful model for studying genetics and health?
Oh, absolutely. So you've got hundreds of different breeds of dogs now, and so each breed has been selected for its own thing, and the breeders trying to fix a certain behavior or fix a certain morphology tend to use line breeding, and so. in these hundreds of breeds, when new mutations crop up, lots of times these mutations then get crossed back to each other. And so you can quickly unmask and you can see what the effect of these new mutations, even recessive mutations, are. And so we have this great model for breeding dogs to perform all of these different things and to look different ways. But it also is this great model for inducing new mutations that cause disease so that scientists can go and understand the genetic basis of these diseases and design genetic tests so that we can avoid them, but also understand the process of that disease, not just in dogs, but in humans.
That relates to human health in what way?
So the fact that we can do translational studies, we can look at dogs and we can identify the gene in dogs that is the defect causing the disease, gives us now a hypothesis in humans. What can we do? Where should we look for the human disease? And a lot of times we didn't know that that disease caused, or that gene was involved in a disease until we found out in dogs, and then we could apply it to humans and improve our ways of diagnosing and also treating the disease.
Okay, great. Now, are there any other things about dogs that make them good models for studying genetics?
Well, it's a lot cheaper to do a genetic study in dogs than it is in people. Dogs live shorter lives. You don't have the same concerns about HIPAA and privacy and all of that kind of stuff. They're more fun to work with, at least I think so.
Yeah, sure.
So in that sense, you can do research on the cheap. If you want to figure out how to make humans live longer, probably the cheapest way to do it is to figure out how to make dogs live longer and then apply it to people.
Interesting. Jacquelyn, anything you want to add there?
Yeah, so we're actually performing our studies using DNA samples collected from the pet population. So these are dogs that are living with their owners. They're naturally developing a disease that affects them in their lifetime. The owner can take them to the vet for a blood sample. They can send that to our labs. We extract DNA, and that's where we're looking for those genetic differences that are driving the diseases. And what's really unique about dogs is they live with us. They share most of the same lifestyle factors and risk factors that we experience. They sleep in our beds, they ride in our cars, they're usually eating our food. A lot of times they share our exercise habits even. Okay. So when you're thinking about diseases like cancer, where these environmental components are really important in addition to that genetic susceptibility, they're really great examples of reflecting what their humans are experiencing as well. And there's lots of examples where dogs who live with smokers, for example, they're at higher risk of different cancers, like bladder cancer is a common one. And that's true for people as well. Oh, wow. And so what we're learning when we're studying the diseases in dogs is relevant to human health and vice versa.
Okay, great. Jess, anything else to add?
Yeah, and so Adam mentioned there are hundreds of breeds. And so the dogs within a breed, these breeds are genetically homogeneous, so they are very similar genetically, and so they share these long stretches of DNA that are inherited together. And in terms of dogs being a great model, these are very useful for mapping, especially variants of large effect size. So we can make use of this kind of genetic architecture of the dog breeds, and it helps us find variants for a specific especially for things like morphology and some disease traits. So not so much the kind of behavior and aging, some of those more complex traits, but yeah, so that's another way that dogs are a great model for.
I'm guessing to take advantage of all that, the power of genetics, you need a lot of samples, so data sets. What are some of the most exciting or important data sets in canine genetics that you've come across?
Right, yeah. And so for me, probably one of the most exciting ones is the data set that my first kind of experience with canine genetics, so as a postdoc. And this data set was published in, I think, about 2016, in that we had over 4,000 dogs, so including purebred dogs, we had some mixed breed dogs, and some village dogs. These are free-ranging dogs that were sampled from around the world. And these dogs were genotyped at about 180,000 sites all across the genome. And so we used this data set to actually find some really exciting associations. So some morphological traits, so things like fur loss. We found a variant that was significantly associated with that. And then in terms of some diseases, we found one of the examples from that is colitis. So this is inflammation of the large intestine. And this particular form of colitis was especially prevalent in boxers and bulldogs. And so we were able to map that to about a 400 kilobase region of the dog genome, and it was very, very significant. And I think also that region has been shown to be associated with inflammatory bowel disease in humans as well. So.
Yep. I teach a class on the human gut microbiome, and I use that as an example. So,
lovely.
That's a great example. Yeah, Jacquelyn, do you use the same data set? You use different ones?
So we use all kinds of data sets.
Okay.
And so in the course, students are going to have an opportunity to work with data sets like Jess is referring to, and also a data set of whole genome sequence that's called Dog10K. This is really amazing because it's actually whole genome sequence, so every single variant in the entire genome from over 2,000 canids. So that includes dogs who represent about 320 different breeds. Wow. There are wolves in there, there are coyotes in there. And so this is all publicly available for anyone to use. And it's great for asking questions about what are the genetic features that go along with canine morphology, like Adam was referring to. What can we learn about the genetics of domestication? We can also use this to think about what's just the common genetic variation that we see across dog breeds. But on the flip side, it's a really useful reference point when we're also trying to find these rare disease-causing alleles that Adam referred to.
Great. Adam, same data sets, additional ones you
Well, I
want to
mean, those
highlight?
data sets are great because of the just sheer breadth and diversity of the dogs that are included in them. I'm kind of partial. There's the Golden Retriever Lifetime Study, which has been put on by Morris Animal Foundation. So this is a data set that's just one breed of dogs, but they've taken over 3,000 Golden Retrievers and they followed them longitudinally through time. So like these other data sets, all of the genetic data is publicly available. You can go on the Betty M. Morris Data Commons and Morris Animal Foundation. You can download the genetic data, but you can also download all of the other phenotypic data that comes from these annual checkups that the dogs have had, surveys that the owners have filled out. There's the potential for access to biospecimens that are collected annually during these visits, and so you can really dive in deeply to ask questions about, why do some Golden Retrievers get cancer and other Golden Retrievers don't get cancer? Does it make a difference if you're a more inbred or more outbred Golden Retriever? And so this is a data set that the students in the class will get to work with to ask things about, the coat color of Golden Retrievers or other phenotypes that Golden Retrievers have and whether we can identify the genes underlying those. And then I guess another data set that's kind of close to my heart that I'm really excited about is so the EMBARK dog DNA test. So this is a dog DNA test that was started at Cornell. I was one of the co-founders of it, and we just wanted to build a better dog DNA test, and so using similar kinds of arrays for what Jess was using for her academic research, we built that into a commercial product, and now we have a database of 3 million dogs that we have all of this information for. And these are not dogs that we recruited prospectively for a study, but it's a cross-cut of dogs throughout America and elsewhere in the world. And we can use that to power. We've had over a dozen studies now where data from these dogs have been used to map traits, map diseases, and the students in the course will have the opportunity to explore some of that data, in addition to being able to test their own dog with an EMBARK kit.
Great. Well, why don't we take a little poll? I think we're going to poll the audience. And have you ever done DNA sequencing or genetic testing for either yourself or your dog? I mentioned earlier that I'd done genetic testing because my mom was adopted. I wanted to know what the other grandpa was like. Found out that I was 25% Ashkenazi Jew. So that came along with some health risks, and that was really helpful to me. So while that data's coming in for answers from the poll, why don't you tell us a little bit more about EMBARK?
Sure. So I'm a professor at Cornell working with Jess and others to do all of this research into dogs, both purebred dogs, pet dogs, village dogs around the world. Lots of people started coming to us and being like, hey, can my dog be part of your study? And can you tell me, what breeds are in my dog or why my dog does this or all of that kind of stuff. And so we realized that there was this need for better kind of research grade dog DNA testing that people wanted. They didn't want it to be just kind of this toy product, but they wanted it to be really scientific. And in addition, we really wanted to have big data sets as scientists so we could start to unlock some of the genes underlying these more complex conditions where we knew we needed tens of thousands or hundreds of thousands of dogs in order to start addressing that. And so Cornell gave us a pilot grant. We got a licensing agreement. We got into the tech incubator here at Cornell. So my brother and I, who had been doing research together, decided to co-found the company.
That's great.
And we launched the consumer product in 2016. So it's been 10 years now that we've been testing dogs in America and around the world, and it's been really exciting to make new discoveries, to have those discoveries go back into the test that you're doing, not just be an academic paper, and suddenly, millions of people have just learned something new about their dog because you have this whole platform.
That's great.
It's been really fun.
Jacquelyn, what's your favorite part about working with Embark?
Yeah, so the platform that Embark uses is actually identical to some of the data sets or data types that we use for our research projects. And so Embark data has actually contributed to many research studies at Cornell and at other universities. And so, for example, my lab studies gastric cancer, which occurs more frequently in certain breeds, like the Belgian Tervuren and the Belgian Sheepdog. That increased prevalence is a clue that there's a genetic component there. And so these breeds are actually also closely related to the Belgian Malinois. A lot of people might be more familiar with the Malinois. It's a little more popular. You might see it as a police dog, a military working dog. And what's really interesting to me is that even though they're really quite genetically similar, the Malinois is not at increased risk of gastric cancer.
Wow.
And so we were really curious, why is that? Like, is there something protecting the Malinois genetically so they don't get this disease? And we were able to take Embark data from dogs representing these three breeds, and we could compare their genomes, and we could ask what is sort of unique to the Malinois, higher frequency there, and is rare in our high-risk breeds. And this actually led us to these two regions of the genome. We were able to follow up and find some variants that seemed like they're in genes that could help protect the Malinois from gastric cancer.
Wow.
And so we were able to use this resource to help us ask one of our research questions in the lab. And so on the slide you're actually looking at, these are our representatives. So this is actually from the Embark data. The plot is called a principal component analysis. This is something that students taking the course will learn how to use and interpret. And so here, each individual dot represents an individual dog. And so their position on the graph represents how genetically similar are they to one another. And they're color-coded by breed, so you can see the Tervuren at the top of the plot. They're all in green, they're clustering together. The sheepdog is in orange and the Malinois is in blue. And you can tell the breeds they cluster with their fellow breed members. But if you were to actually zoom out, if you were to take maybe some boxer data, chow chows, Scottish terriers, breeds that are not really closely related, what you'd start to see is that these three breeds that are close relatives start to kind of merge together. So it sort of depends on your perspective. But this is one of the ways that we can think about as scientists, what are the relationships between individuals that we use in our studies.
Yeah, I think it's a great way of reducing the complexity of the data. We talk about this in my class, just data reduction is so important to be able to see a signal in all of the genetic noise. Jess, how do you use the Embark data?
Well, actually on a bit of a personal note, so Blueberry here has an Embark profile. So I think there are, Adam can correct me if I'm wrong, but there are about 11 or 12 golden retriever specific health traits.
Yes.
On the, okay,
Yep.
but Blueberry, can you sit? And so things like ichthyosis, which is a dry skin issue. There's a bunch of eye diseases, so things like progressive retinal atrophy. And so he's clear for all of those. But one thing that he does have is he has one copy of the ALT allele. So ALT is alanine aminotransferase. So this is a liver marker. So when you have elevated levels of ALT, this kind of is indicative of liver disease or injury. And it's the same thing in humans, right? So just in the blood chemistry panels. And so because he has one of these risk alleles, that means that his normal ALT levels are a little on the low side. Yeah, so another thing that he, another thing that Embark shows is the coefficient of inbreeding. And I think we have a plot here to show that. So this is his coefficient of inbreeding was 21%. So this is kind of a measure of how related a dog's parents are. And so obviously the higher the percentage, the more inbred the dog is. And so you can see Blueberry is 21%, and the other blue lines there are how he compares to other golden retrievers.
Great. Now, how would an owner use that kind of information if they wanted to know something about their dog or their dog's parents?
Yeah, so I think it's interesting. It's more for, I think, if you're going to breed your dog, which we're not doing with Blueberry, but yeah, if you're a breeder, that's very important information you can use to choose who to breed your dog to for the next generation, because you don't want to get too high on that graph.
Well, and Blueberry's breeders, so you have some idea of Blueberry's family medical history, but people who are adopting a dog from a shelter really have no idea.
That's
and
true.
So not only learning the breeds of dogs and learning the specific known genetic defects, but this opportunity to find relatives of your dog and then talk with them to see maybe they have a similar health concern about their dog and they know something about it.
Now, how complicated is the process of actually getting the testing done?
Yeah, and so as Adam mentioned, so students enrolled in the course can submit a swab from their dog for embark genotyping. So hopefully Blueberry is going to demonstrate how easy this is. Can you sit? Good boy. So we have the embark kit here. And we're going to wander away.
We should make it bacon flavored.
Yeah. So it is really pretty easy, and there are lots of instructions. So do usually say no eating about a good half hour
About
or so
half hour.
before we take this swab. So Blueberry, can you sit?
And it's not that the DNA on the food is going to contaminate the sample. It's just if there's like chunks of food, it's going to clog the tips when we're trying to extract the DNA.
Yeah, so I'm just trying to get it kind of in the pouch of his cheek there. I'm just going to rub gently on his cheek just for about 30 to 60 seconds. I think longer is better.
It's a sponge that's absorbing the saliva. It's not, you're not like scraping you're not trying to get blood or cells or anything. Just what's in the saliva.
I'm going to go the other side. Right. Okay. I think that's about all it's going to take for now. And then just unscrew it and put it in the blue liquid. That's it. Yeah, so it is relatively easy. Give it a little shake.
Give it a little shake. There you go.
Good boy, Blueberry. You want a bean? Good boy. Okay.
Great. I think one of the exciting things that comes from having publicly available data is the opportunity for citizen science, and that is a good segue to talk about the course. So let's talk about the course a little more. It's called Genomic Data Science in Dogs. Can you introduce it and what the purpose is and what students can expect to learn from it and do with it?
Yeah, so the course is really designed to teach the data science skills that we're using all the time in our labs to ask these genetic questions. And so the students are going to get a background in genetics from the perspective of the canine data sets. They're going to learn a lot of the fundamentals of data science in general, so these can be applied across any sort of data set. So they'll learn about data cleaning and normalization, visualization, and really what we want is for the students to have, not necessarily every genomic tool that's out there, but kind of the fundamentals so they can start asking questions on their own. They can take a public data set and they can ask things where they can gain some insights about, maybe some basic mapping analyses or finding causal variants and understanding that process. And so that's really the goal throughout the course. And it's designed to be completely asynchronous, so it really allows for flexible learning. There's 4 lectures per week. And then there's a tutorial that's designed to help the students gain the skills they need to do the lab exercises. And so that's the point where they're going to apply what they've learned that week to an actual data set.
Great. Jess, anything to add?
Yeah, so this course is geared towards undergrads, but we've also made it accessible for pre-undergrads, so high schoolers, because we were aware there's an interest in some of those high schoolers wanting to get their hands on some cool data sets and learn some of these things. And as I think it's been mentioned, this course is fully asynchronous. So if you live in another country, like New Zealand, then you're able to enroll and take part. Yeah.
No, I think that's great, and I think it's good to mention the high schoolers. I did some independent study with a high schooler during COVID, and we accessed a publicly available data set for human microbiome stuff, and they were able to use that project as part of their application to college. And so it really is a great opportunity to use tools. I think they're very amenable to anyone with basic math they got in high school, and but you can use that to answer your own questions, and I think that's really exciting. Adam, anything else you want?
I mean, people love dogs. There's lots of interest in dogs, and to understand dogs, you really need to understand dog DNA. And there's also lots of interest in data science. People want to build skills around data science, not just in a genomics context, but just in general. And for me, I'm not a textbook learner. So I'm going to learn something when I care about something and I'm given the tools to kind of do it. And so to me, there's this sort of synergy where, hey, we can teach you a lot about dogs, but you don't have to take our word for it. We're also going to give you the skills so that you can explore real data sets about dogs and see how those conclusions are made, and maybe ask your own questions with them. So we're not, if you want to become the world's next great canine genomics expert, great, but this is building a toolkit that's going to be useful in lots of different domains, and hopefully in a way where it's resonating because it's something that the students care about instead of just textbook examples of how to do data cleaning and data analysis.
Yeah, once you have the DNA, the tools are pretty much the same whether it came from a microbe, a dog, or a human. So I think that's really important. So I think we have a question from the audience. So Matt from New York City asks, will this course teach us skills that we can apply to human genetics or other fields? So we touched on this briefly, but do you want to just expand on that?
Oh yeah, I mean, absolutely. So the A, C, T's, and G's are the same between dogs and people. I mean, we teach you some of the dog specifics, so you know that dogs have 39 pairs of chromosomes, humans only have 23 pairs of chromosomes. So in some of the specifics, it's going to be a little bit different, but you're using the same tools. You can analyze the data sets in the same way. In R, you can do mapping studies, you can do visualizations, you can use principal component analysis. You can organize your data the same way. So you can hit the ground running with whatever species genetic data of interest you have, but we've shown you where, what are the specific things you need to know about dogs to analyze that, and now you know what to ask about the organism that you're looking at. And in fact, I think you're actually going to be a better human geneticist if you start out with dogs, because you're going to start out knowing to do comparative genomics, right? We're teaching you how to use the browser, we're teaching you how to compare human and dog genomes. That's a skill that's going to serve you well, no matter what organism that you're studying.
And you're a better human if you like dogs. So.
That's right.
There's that. You mentioned a couple of pieces of software or programs, so let's, I think we're going to poll the audience one more time. So do we have a poll on the use of R? Where did I've lost my notes there. Where did it go? Where's our poll? I think we're, so let's take R for example. What's your experience with R? Have you used a program like this before? Are we loading that up? I probably butchered it. Okay, how much experience do you have with the program language R? There we go. So while we're waiting for that to come in, I'm gonna just ask you a last question. What do you hope students take away from the unique Genomic Data Science in Dogs course?
Well, obviously, I hope they take away 3 credit hours from Cornell. We've designed the course so you can take it for letter grade, or you can take it pass-fail, so you can take it at whatever level you feel comfortable at. But ultimately, it's really, I want them to take back confidence, right? Like, oh, I can actually deal with real data sets. I know what I'm supposed to be doing. I know how to ask interesting questions. And I know enough about the context of the data that I can formulate interesting questions. And so just to have that confidence is really the difference between somebody who maybe just has textbook learning about something in theory and somebody who's actually rolled up their sleeves and known how to work with real data.
Yeah, I think that ability to pose a new question is a great way to demonstrate mastery of the material, and so I think that's great. Jacquelyn, anything else to add about the course?
Yeah, so they're going to learn a lot about canine genetics specifically, but importantly, they're going to learn a lot of fundamental genetic concepts. And so as we've touched on, those can really be translated over to any species and applied in different systems. And they're also going to learn really the fundamentals of data science. And so this will serve them well in other areas and other species.
Jess, anything to add?
Yeah, so I think we hope that they will, students will gain a deeper appreciation of the genetic variation that is available, I guess, in dogs, and in that way make them kind of appreciate some of the characteristics and the traits that they see in their own dog and makes their own dog unique.
Yeah.
Yeah.
That's great. I think that's really important. I think connecting what they see with what we understand about genetics is such a powerful tool. Now, just looking at the poll, I see that our most common response is what is R, and that's okay. I think that's important to point out that you don't need to know what R is to take the course. These are just software packages that are publicly available, and they just give you a tool basically to go into toolbox.
And that's why we provide tutorials, so you can get training wheels on before you're sort of examining that data set yourself in the lab. And so our hope is to take students who really don't have the experience yet, give them that experience, and then have them, let them loose on real data.
So no experience required.
Right.
Great. Okay. So I think we have one last question from Megan in Dallas, and she's asked, how can understanding dog genetics help us better care for our pets?
Yeah, so this is a great question. This is something I think about a lot for our cancer studies and these complex disease studies, where as we learn more about the genetic variants that are really driving these traits, this is information that would be really useful for your veterinarian to know. So if you find out that your dog is high risk for a particular disease that's later onset, maybe you start screening for that earlier. A lot of times in canine medicine, we have a hard time with, diseases might be diagnosed really late because the symptoms are just not obvious right away. And so if you can catch that earlier, now you have the opportunity for treatments to potentially be more effective and improve the outcomes for your dog.
That makes sense, and it tracks with my experience. When I got my genome done, I went in and talked to my doctor because I have a mutation in an enzyme that would metabolize warfarin. If I got a regular dose of warfarin, that might be bad for me. So I think there's a great and the doctor didn't know what to do with the genetic information at the time, but having those conversations, I think, are really important. Jess, anything to add?
Yeah, so actually I have that same warfarin.
Oh,
yeah.
cool. New Zealand.
Yeah, there we go.
Are you relatives?
Well, maybe. Yeah, so just kind of building on what Jacquelyn said, so understanding the genetics, or the genes and the genetic pathways that are involved, so the kind of the genetic mechanism of how a disease develops can help us as researchers to potentially design better treatments and better therapeutics, better therapies for treating dogs. So, yeah.
Great. Well, Adam, you want to last
Yeah,
word?
I mean, I was just going to say, like similar to warfarin in sensitivity in humans, dogs have multi-drug sensitivity. So about 1% to 2% of dogs are carrying a mutation that leads to high levels of toxicity when they encounter certain drugs or certain toxins in their environment. And it's really valuable for owners and veterinarians to know about that and a bunch of other risks. And I think breeders have traditionally known to do genetic testing for the things that are found in their breed, but it's now that we can offer tests for all breeds at once through these array panels that we do, that people who adopt dogs and they don't even know what the breed is in the dog can now test for everything that might be in there, because the dog is a mix of all these different breeds, as well as, of course, figure out which breeds are in their dog.
Yeah, bring you peace of mind, or allow you to be proactive.
Right.
So that's great. Well, wonderful. Thank you all. It's a great message for us to end on. Again, anyone interested, you can apply for the summer course by visiting the link in the chat. Unfortunately, that is all that we have time for today, and I just want to thank our faculty, Drs. Boyko, Evans, and Hayward, so much for joining me today. And it's great to be here with you, and I'm grateful for the chance to have joined you there. So a reminder to register for the course. It's open now, and we look forward to seeing you all in the class this summer. Thank you, and have a wonderful afternoon, everyone.
Thanks, Anthony.
Thank you.